For example, in the cell or during the cell division, for example in the metaphase stage, they are the chromosome which is present in the metaphasic plate. So now the spindle fibers are formed here. So this structure is called as the centrosome. So now what is the centriole, what is the centrosome? So now if we see the structure, the centrosome, here it looks like this. So this is the centrosome. So in the centrosome, two centrioles are present and they are present perpendicularly to each other. So they are called as the centriole. So in one centrosome, two centrioles are present and they are present perpendicularly to each other here. And besides this centriole, some amorphous structure is present and they are called as the PCM. PCM means pericentriolar matrix. Just imagine, or just like the cytoplasm. So that means inside the centrosome, two centrioles are present and they are present perpendicularly to each other, and now the PCM or the pericentriolar matrix are present. So now if we look at the centriole structure, let us suppose this is one centriole. So in the centriole, the structure looks like this. Just imagine. So that means in the axoneme, two structure are present like this here. So that means, for example, this structure, that means one tube and it is joined to the another tube. If we take cross section, it looks like this. So here three tubes are joined to each other, 1, 2, 3. So just like that, 9 peripheral structure, 9 peripheral structures are present. So in the axoneme, this is the axoneme structure. So now in the basal body, it looks like this. That means this is the axoneme structure, or this is the basal body, which is similar to the centriole. Okay. So in the basal body of the centriole, the same structure like present, but what is the difference in the axoneme? So the peripheral link are called doublet. So they are the doublet. That means they are present two ring structure, but they are the triplet. So that means they are present or the peripheral link same. It is called as the peripheral ring structure, and this is called as the middle part. They are called as the central ring structure, or the central link or central structure. It is also called as the hub. So in the peripheral ring structure, the triplet microtubules are formed, while in the axoneme structure, the doublet microtubule structure are formed. And another difference is that inside the central region, there is no microtubule structure, but in the axoneme structure, they have two singlet structure are present. So that's why the axoneme structure is called as the 9+2 arrangement, and the basal body or the centriole is called as the 9+0 arrangement. And here the difference is the 9 is the 9 are the doublet, and here the 9 are the triplet. These are the main difference between the exonym structure and the basal body structure. So now look at the structure here. So it looks like the bullock cart wheel structure. That means in the, let's suppose this is the bullock cart, this is the one wheel, and inside it another wheel like structure. So here the centriole is also looks like same as bullock cart wheel structure. So that's why the centriole is also called as the bullock cart wheel. This is the very important, and in the previous exam this question was arised that which structure is also called as the bullock cart wheel structure, and the answer will be the centriole. So this is the all about the structure or the difference between the exonym and the basal body. So the basal body, it is same as the centriole as in the basal body structure. Here the centriole forms the cilia and the flagella, and the process is called as the ciliogenesis. So now this is the centrosome, this is the another centrosome. Here in the centrosome two centrioles are present perpendicularly, here also two centrioles are present perpendicularly. So now actually first in the cell one centrosome are present, but for example in the cell cycle, this is the M phase, this is the G1 phase, this is the S phase, this is the G2 phase. M phase means division phase, that means mitosis or meiosis. G1 means this is the gap 1. This is the S phase, that means here the DNA is synthesized or the DNA becomes double. So now this is the G2, that means gap 2, that means the gap between mitosis or the meiosis and to the synthesis phase. So during the gap 2 phase, so now the centrosome becomes the double. So the centrosome actually in the cell the centrosome are present 1, the number of the centrosome are 1, but during the gap 2 phase the centrosome becomes double, just like the binary fission. So in the gap 2 phase it becomes the double, and now it comes to one end and another is come to another end, and now the mitosis and the meiosis occurs. So now the spindle fibers are formed. So this is the all about the centrioles or the centrosomes. So now come to what is the function of the cytoskeleton, or what is the function of the microtubule. As I have already discussed, but just as a summary I am telling you that one is the spindle fiber formation, the second one is the formation of exonym and basal body of cilia and flagella. Third one is the it also helps in the cell differentiation. So that means which cell should convert to which cell, and it is determined by the microtubule. Another is the it also distributes distributes to the cell pigment. That means, for example, the chloroplast or the melanocyte, and another is it also helps in the cell movement. That means during the gastrulation, one cell goes to another, or one cell again come to here. So this is the cell movement, and this cell movement is occurred by the microtubule, and most importantly it provides the support to the cell or to the organisms. So this is all about the structure of the microtubule. So if we like the video, then please like and subscribe my video. Thank you.